Good morning, everyone. Welcome to our webinar, What Does the Research Tell Us About Cover Crops and Soil Health? I am Rachel Cochran, Water Quality Extension Associate for Paulding, Defiance, and Van Wert counties. And we are joined today by Bridget Moneymaker, who will be doing the Q&A at the end of the session. She is a Water Quality Extension Associate for Always, Allen, and Mercer counties. And today's speakers are Elizabeth Hawkins, OSU Extension Field Specialist in Agronomic Systems, and Steve Culman, OSU Soil Specialist from the School of Environment and Natural Resources.
Good morning, everyone. Steve and I are really excited to share with you some of the work that we've been working on here at Ohio State over the last few years to help bring more information about soil health to folks in Ohio and even more broadly than that. So a few years ago, 2 years ago, we started, well, let me step back a second. 5 years ago, we started our eFields on-farm research program. And this is a program that we wanted to bring field scale on-farm research to farms to help generate information that you can use to improve your operations. 2 years ago, the question was brought to us about soil health specifically, and how could eFields help improve soil health or collect data to help inform us on how we can improve soil health. We do have a lot of other information in our annual report. The 2021 report has over nearly 250 trials reported. You can scan the QR code on your screen here, or you can request a printed copy through your local extension office. Towards the end of that book in our soil health water quality section, you will be able to find some of the data that we talk about today. So again, the eFields program, what really makes it special is that we try to keep it driven by farmer questions. Some of the things you guys are dropping in the chat, I'm going to make notes of, because we want to be sure that we're focusing on things that provides information that you can use in your operations. So whether that's soybean seeding rate, fungicide, or soil health, we really want to be looking towards things that matter to you. So when we looked at the soil health project, some of the things that we wanted to better understand was, how does soil type influence soil health values? We know that as we look across that range of textures, the metrics may be different. One question we got was, how much does sampling depth matter? You take your standard soil health or your standard fertility core, usually at around 8 inches. But a lot of people were talking about, for soil health, should we sample more shallow? So we'll talk about the methodology here in a moment, but that was a question we were trying to strike out to answer as well. We also wanted to know how some of the conservation practices that are being conducted on farms currently impact soil health metrics over time. And as part of that, we wanted to help you set baselines so that you can track soil health progress at the field level on your farm. And specifically, I think Steve's going to spend a lot of time on this today. What is a good soil health value? What's a good starting point? And what kind of changes can you expect to see over time? So here's just a quick overview of what our sampling protocol looked like for this project. We wanted to use a whole field sampling strategy. So you can see there on the right, zigzagging across the field to make sure that we're getting a representative soil sample of that whole field area. And then we're aggregating 10 to 15 cores of soil into that sample. To answer that depth question, we collected samples at 3 different depths: 0 to 4 inches, 0 to 6 inches, and 0 to 8 inches. And we kept those samples separately. So each field, we submitted 3 different aggregate samples for analysis. Over the 2 years, our timeline for sampling, we tried to sample between May and July and keep that sampling time point consistent over the years that we've collected data. To answer the management question, we focused in on 4 different management schemes that we wanted to be able to classify our samples into. So fields that had been in long-term no tillage, conventional tillage, fields that had been cover cropped, and fields where organic nutrient applications had been made, AKA manure. And we also, in addition to just classifying these, tried to quantify how long each of those fields had been under that particular practice.
Great. So thanks, Elizabeth, for that intro. I'm going to take over from here and just thank everyone for joining this morning and talk a little bit about our methods. And really what we've done the last couple years now has been consistent, which is pretty nice because although we haven't really done much of this at this point, we are able to combine the data over the last 2 years now. And so today I'll just be focusing really, we presented 2020 data last year. We're going to present 2021 this year with a little bit more. And there's a lot of commonalities, and we'll talk about that, which is, it's been pretty interesting to see. So you can see the map here in terms of the shaded counties where we were in. About 120 fields. There's a few more than that, actually. We had some stragglers and whatnot, but again, sampling at that 0 to 3 inch depth, or the 3 depths rather. We ran a routine soil test with each one of these samples. And then last year we focused on permanganate oxidizable carbon, or POX, and aggregate stability. This year we've expanded the measurements with support with the Ohio Soybean Council, checkoff dollars to help support these analysis and some of the logistics in terms of pulling off a project like this. So we've added respiration and soil protein. And for today's talk, I'm really just going to be focusing on the methods here. So some of the routine stuff, pH, organic matter, cation exchange capacity, and then these 3 soil health indicators that we've talked a little bit. If you've joined webinars in the past, we've talked a little bit about these, but for those that might be new, I want to just kind of mention what these are. Okay, so we'll start with, in some ways, I consider a breadwinner test of soil health. It's this funky word, POX. It's not an affliction. It's just a fairly simple, cheap test to measure this kind of active carbon. So if you've ever looked at, so POX and active carbon are really the same test. It focuses in on a biologically active pool of organic matter. So we measure total organic matter with most routine soil tests, but this is really getting at what we would consider a very active fraction. There's a lot of the total organic matter that might not be plant available, or might be in a very stable form. This POX is a little bit more biologically active in that regard. And so at the end of the day, it's a very sensitive indicator to management, especially when compared to total organic matter. And that's why we really like it. It's a simple test, and time again, not just in Ohio, but in other regional or national studies, POX has been shown to be a very sensitive indicator. Okay, so what is it? It's this. It's kind of an amorphous pool of all the good action that's happening in the soil. We know functionally it represents what we would consider a microbially processed pool. So it's not recent inputs, it's what microbes have chewed and have gone through kind of a microbial decomposition process to some extent. And that really means that it's more a mineral associated fraction of carbon. That's what we would typically consider a little bit more permanent. So when I say mineral associated, I mean things that are making these linkages with things like clay in the soil. And so that's really where organic matter, the kind of the ground zero of where organic matter is built. And so we think of POX as a very nice indicator of building organic matter. Okay, so this is POX in a nutshell. We move on to the next test, which is respiration. And this is simply measurement of how much CO2 is released after we take a dried soil and add a certain amount of water. So this is really the soil breathing, the microbes coming back to life, and then how much CO2 or carbon dioxide that they respire off. And we measure that the concentration of that carbon dioxide in an incubated little chamber here. Okay. We know that respiration reflects, in contrast to POX, respiration reflects more recent additions of carbon into the soil, things like crop residue, cover crops that are not as decomposed, okay? So, and because of that, we believe that respiration, we've got lots of evidence and data that we've generated to show that it's a better indicator of kind of nutrient availability or what's immediately available to plants, okay? And I just mentioned this because a lot of folks have, if you've had soils tested for, sent it to commercial labs, Solvita is the kind of the commercial implementation of a respiration method, but labs can run this a number of different ways. And we don't use Solvita, but it's measuring the same process, okay? And then finally, well, let me just talk a teeny bit about this, just to give you a better sense of POX versus respiration. Okay, there's a lot on this slide, and it's not terribly important, but we can think about kind of conceptually pitting POX and respiration together, using them in conjunction to get some idea of what's happening in our soil. Okay, and so I just told you that POX is a good indicator of organic matter storage or building organic matter. Sometimes we call that stabilization, carbon stabilization, if you will. And then respiration down here is an indicator of nutrient release, okay, or mineralization. And so, thinking about organic matter, you can either be building it, stabilizing it, or you can be burning it up and releasing. And we want really both in a high functioning soil. Okay, so we want processes that are building organic matter, so we're not completely depleting it, but in the immediate sense, we want some mineralization happening, so nutrients are flowing and crops are able to take that up. And so, again, conceptually, if we kind of pit these against each other, we don't want all storage and no feeding crop, and we don't want all feeding crop and no storage, because we're going to run the tank empty. So we'd like to think about getting systems in kind of at least agricultural systems in this high stabilization and mineralization piece. Okay, here's just some data from Michigan to kind of show that those these long-term studies, these kind of concepts do bore out in the real world. Okay, so just thinking about POX and respiration, I think that they're nice complementary indicators that really reflect different processes that are happening in the soil. So because of that, we measure both, and we think that they're both important. Then the final thing is this soil protein. And protein, of course, we think about protein and nitrogen a lot, in modern life, that those things are related with foods and whatnot. But of course, there's a lot of protein, both plant and microbial protein, that happens in the soil. This is an organically bound nitrogen. So when unlike nitrate or ammonium, which is just kind of an inorganic or a free nitrogen, this is actually bound with carbon, okay? And we know in soil and studying soil dynamics that the supply of amino acids or proteins is often the most rate limiting step for soil nitrogen cycling. So if we can quantify the pool, the source of nitrogen that actually is being released, then we have some indication of nitrogen availability. Okay, we know that it's a more stable pool, that is, it's less ephemeral or fluctuating. We can go out in soil in the springtime, in particular in Ohio, and measure ammonium and nitrate 1 week, and then go back and measure it the second week, and those values can be drastically different. And so we believe, as an indicator, protein is a really nice, it's more stable and therefore a little bit more useful from kind of a longer term planning perspective, like a whole growing season, or say multiple growing seasons. We've got, in terms of the function, we've got evidence to show that soil protein is an indicator both of nitrogen storage, that is actually kind of depositing nitrogen, building up that savings account, versus nitrogen release. And so we're still trying to kind of sort out a little bit more about what protein what it reflects and what actually influences it. So this is, of course, an active area of research and a work in progress in terms of what we're trying to do. So anyway, suffice it to say that those three indicators, this is what we've been focusing a lot in measurement, and the last couple of years we've run these things we believe are complementary. You've likely heard about carbon and nitrogen in soil and how that really controls organic matter cycling, right? And so we've got really conceptually our POX, or active carbon pool, our protein, or our nitrogen pool, and then respiration, which kind of integrates that and feeds off these things. So we believe kind of conceptually that these are three complementary indicators. Okay, I'm going to leave it at that and move on to some of the results. All right. So last year we looked at this and thinking about the influence, the role of soil type on soil health. And we know that that different soils across Ohio, of course, matter, and the management of the soils differ because of inherent constraints and what we expect from a given soil changes across soil type. So I'm just using a very convenient indicator of soil type, which is cation exchange capacity. And we can look at things like organic matter. This is total organic matter now, loss on ignition organic matter, versus this POX and respiration. And what you see that, and this is commonly found, is that POX and total organic matter are more closely related than respiration. This blue line here just indicates the kind of the best fit. So it's a trend line, and we can kind of look to that. We see that organic matter has the steepest slope of any of these lines, POX the next, and then respiration, it's not really, the slope is much more flat, and there's a lot more variability. And this is kind of what we like to see in some ways, that the variability around these lines, because it means that the soil health indicator isn't only controlled by soil type. We know that as CEC increases, as our soil content of clay and organic matter increases, this is primarily what drives CEC, is clay content, a little bit of organic matter. As that increases, so do our soil health indicators. And these are 2 carbon indicators, okay, indicators of organic matter. But then there's some variability around that, which just indicates to us that there's a lot more controlling respiration values and POX values than, say, total organic matter. Okay, so this is again a similar finding that we found last year, which is a nice affirmation that spending the time to measure additional things like POX and respiration protein are actually worthwhile and provide additional information. Elizabeth talked about in the beginning about this, this depth influence, and not surprisingly, if we sample soil, say 0 to 4 inches or shallower than a true 0 to 8 inch sample, we're typically going to get higher numbers. And this bore out with, not surprisingly, with our soil tests. And so we can look at our 3 depths. Now, this is average across all fields, so it's just a general average, a statewide average. And we could, can and have gone back and looked at things, break them down by region or what have you. But in general, or really universally across the board here, as our surface samples have return higher values than our lower, samples that we've sampled to a lower depth. Okay, so 0 to 4 returns higher values than 0 to 8. This is important because what we're going to be able to do at this point, after 2 years and probably close to, we've got about 450 soil samples, maybe 500 soil samples at this point across the 2 years. We're able to combine this information and develop some relationships. So if you sampled at 0 to 6, we can generally convert that to what a 0 to 8 inch sample is. So this is some nice information, just practical information in terms of being able to relate soil information that was sampled at a different depth than 0 to 8. And of course, we've done all of our sampling at a 0 to 8 inch. That's because that's what the tri-state recs were originally based on. We did not change that. And the justification is that's typically the zone of mixing, for a kind of a classic chisel till or a plow till. Okay, so there's a lot to be said about what depth we should be sampling on. We'll hold that debate for another time. But if we sample at 0 to 4, 6, or 8, we should be able to be on the same page in terms of what our values are at this point in time. Okay. So I want to talk a little bit more, just get into some of the influence of management. This is, of course, a big interest. And I'm not showing, I could show you other values. I'm really focusing on POX and respiration for this. I could show you soil protein as well. But this is just thinking about manure and the role that manure plays in building soil health and crop nutrition. These are just distributions, okay? And so how we read this, we've got our value. This is percent organic matter, POX, and then respiration, okay? And then we've got these kind of two different curves. One is manure, fields that have not been manured, and fields that have received manure. And how we read this is basically as this curve increases, that means more observations. So this is, again, integrating across all of our soils that we have. And as, so around for manured samples, this green or bluish, most of the observations, fell around, say, the upper 2s to 3% organic matter, where fields that have not historically received manure, maybe the majority of those fields are around 2.5%. But importantly, not just the absolute peak, but the entire distribution is essentially shifted right or shifted greater when in this blue samples, that is samples that have received manure. And that's true across our respiration and our POX measurements as well. So, we know that manure, of course, has wonderful benefits for building soil health, and this is some proof of that across the statewide. Okay, let's talk about no-till and how years in no-till influence soil health. Okay, and so this is an interesting finding, and it's not what we expected. It's certainly not what I would expect, but we can see years in no-till and the influence of it on organic matter and the influence on POX and respiration. Of course, these trend lines are not trending as we'd expect. And of course, there's a lot of variability around here, okay? So this is pretty much exactly what we found last year with this data. And, it's not to suggest that no-till has no role in building soil health. It just means to me that it's more complex, that it's not, the relationship needs to be looked at on kind of an individual soil basis, or maybe across the state, no-till influences things in different ways or different soil types. So it's not just a slam dunk, at least from the surface here, that if you're in no-till and have been in no-till a long time, you're generally trending towards greater organic matter and greater POX and respiration values. We can contrast that with years in cover crops, okay? So here we actually looked at both cover crops and in perennials, because we had a number of sites that were in say permanent pasture or hay sites here. And so we looked at this in terms of kind of combining this variable of cover crops. So thinking about continuous cover cropping versus or being in perennials in that rotation, okay? And so here we can see much stronger, I know we've got this, kind of values that are out in the 25-year mark, but even if we take that out and just look at what's happening, say over the first 10 years of this, we see positive trends in both organic matter, POX, and respiration. Again, these aren't slam dunks either, but they're a little bit stronger than we saw with no-till. And that's the same story, interestingly, the same story that we saw last year as well. So the 2 years of data really kind of line up and are very, have a lot in common with each other. So to summarize here, of course, soil type matters. There's a lot to be said about this, but similar to something like a soil test K value or whatever, we need to really know what type of soil we're on and kind of calibrate based on that. So as C&C increases, we saw that our total organic matter, basically everything I showed you here, increases as well. Depth of soil sampling matters. This is basic agronomy. Folks know this. When we're trying to pull soil samples, we need to keep depth consistent, or we're really going to be muddling our results and kind of making the soil values, the soil health, whether they be a routine soil test or soil health value, we need to keep depth of sampling consistent. Management matters, right? I just showed you some years in no-till. Had both increasing and decreasing soil health values, and years in cover crop or perennial, we really saw more positive increase, positive trajectory. Okay. Then I'll just, this caveats that we're just in some ways starting this on-farm work. Much more work is needed. We're going to be combining data from both years and making sense. I think moving forward, the depth question is likely been answered, and we'll talk a little bit more about kind of next steps, but it's good to have data and kind of base some of this on, okay? So I'm going to talk a little bit about a question that we get all the time. We run soil health tests, and then, growers, one of the first questions, well, what's a good value, right? So like, you gave me some numbers on a handout. What does that actually look like? And so again, the soil type is important, right? And we need to first calibrate by soil type for us to give you, to answer this question essentially. All right. I'm going to show you, this is data from a project that I'll talk about a little bit after, but this is this idea that we can take coarse, medium, or fine soils, or that is sandy, silt loam, or clay loam soils. Fine soils would be heavier soils or clay soils. Coarse soils would be sandy soils or light textured soils. And when we graph these or think about how these distributions behave, this is a unique, an organic data set that I'll talk about here in a second, but we can see the same thing, that heavier soils or these clay soils, fine soils, have essentially greater values relative to these coarse soils. So again, the whole point here is that we really need to kind of calibrate what type of soil we're working on. If you're in a Hoytville clay, it's going to be a whole lot different than a can-filled silt loam, say here in Wayne County, okay? So these are our values, and for what it's worth, we've taken and we've kind of created these distributions and then basically made percentiles. So we sampled, all these soils across farms. We've classified them into a sandy soil, a silt loam, or a clay loam soil based on CEC. And with that, we've said, here's the bottom 25, here's the 50th percentile, or the middle number, 75th, and then the 95th would be considered, the A+ students or what have you. These are the ones that are the really high values, okay? So I'm not expecting you all to retain all of this information, but we'll be publishing this and pushing this out in the next couple months, and just to give some baseline idea of like what a good value is. So here's a sand. Okay, I'm going to show you the same slide for a silt loam, but the numbers change. And then I can show you the same slide for a clay loam. So CCs of 18, say to 25, we've excluded muck soils from this analysis because muck soils are their own animal by themselves, okay? So again, I'll just flip through back to sand, and you can look at some of these values, and you can see that they're increasing from sands to silt loams to clays. One last time, because it was so much fun the first two. Let's do it one last time for those that are interested. Sands, silt loams, and clay loams. Again, happy to share this information. We're going to be putting this out more. Okay. And I want to just talk a little bit to give you all a sense of kind of where we're going with this research. We've got a number of projects over the last 5 years that we've measured soil health in different aspects on different farms across the state. We probably have about in total a little over a couple thousand soil samples that we've sampled on farm. And we're in the process. It's kind of a big job, but it's like pulling all this together and really trying to address some of those questions that Elizabeth raised in the very beginning, particularly like how is management impacting soil health, and how do we get there? Like, how do we get to soil health faster? What's really effective in terms of helping us get there? And that's a kind of a weighted question to begin with, because it really depends on what you're trying to do, what you're trying to accomplish. And just saying, I want to build soil health is such a kind of a vague goal. We really need to drill down better than that. Okay, but anyways, I digress. And let me just talk a little bit about a research project. This is from USDA organic funded proposal. It was a 4-state survey of looking at organic corn farmers. Okay, so it's an organic specific project. All these producers were certified organic, all growing corn. The results are published. Christine Sprunger, who's a professor here at OSU, the results are published. We're happy to share that information with you as well. Some of the questions were what's the best practice and what's typical soil health values? This was all done through the mail. So, we sent out surveys asking if growers were interested in a free soil health test. They brought us, we gave them postage, they shipped soils back, they sampled soil, shipped it back, and filled out this management information. It's pretty similar to the same process that we did with the e-fields, just little nuances here and there. Okay, so the shaded counties here are where we were at. And we looked at crop diversity. Organic farms are typically, we might think of them as, farms that have greater soil health because they do things, differently. And typically organic farms have more crop diversity, whether that be through cover crops or cash crops or what have you. And we looked at crop diversity versus some of these soil health measurements, right? So I'm showing you POX, mineralizable carbon. This is our respiration value. It's the same thing, just a different word, different name. And then soil protein. Here's total organic matter, then total carbon and nitrogen. Okay, so these are really getting at the same thing, but just measured a little bit differently. What do we see? We see actually a negative decline with crop diversity and soil health indicators. And we were like, what is going on here? This makes no sense. You're telling me, as we increase the number of crops, and we had to, I'll just say this is an index. So we basically quantified how many crops they had grown over 4 years. But what we see is that it's trending down. And again, this is not what we expected, totally counter to what we expected. And what we found then is looking at tillage frequency. And again, we asked about tillage practices. And so tillage frequency here has a big role in terms of what it does to soil health. And as that tillage increases, our soil health is, typically declining or flat, certainly not improving. Okay, so I'm giving you the abbreviated CliffNote version here, but what we didn't really account for is perennials. And a lot of organic growers do have perennials in rotation and use that as kind of a break to build fertility or to get weeds under control, or both. And what we found was that years with a perennial present in that crop rotation, whether it be 0 to 4, had big influences on kind of soil health across the board. Okay, and so the caveat here was crop diversity. The more crops that were grown, the less perennials were. And because there's a lot of crops and it's an organic rotation, there's a lot more tillage that occurs. Okay, so again, glossing over some of these data, But again, what we found was that, management is complex. Certainly with organic growers, it is complex. And that crop rotational diversity interacts with tillage in organic systems to either improve or essentially degrade soil health. And so what we really found was with crop diversity, as number of crops increased, our soil, the tillage in these organic systems increased, and that had a negative impact on soil health. But when we included and looked at perennials in that rotation, these are typically alfalfa or hay or, grass-based number of, but we could just call it hay essentially. As hay was in that rotation, the tillage decreased and was associated with greater soil health. Okay, so again, this is just a simple kind of example that we're done with this project, but it really, for us, highlights the importance of conducting on-farm research and really trying to kind of tease apart some of these relationships. Okay, and doing it when we have, like we're doing with this eFields project, the kind of the breadth of this, doing across so many farms across the state, and you get all those different managements and there's different soil types and the different crops that are grown. It really provides a very, very robust kind of framework. It's messy data, but it really helps pull out the really important and strong signals out of that data set. Okay. So again, what we're going to do, we're going to take data sets just like I showed you here. This is organic specific, but we have a lot of data that are from conventional, ground, and we're pulling those together again to provide, continue to, I'm going to skip back on you all a little bit, taking data like this where we have like what's a good baseline data set, kind of filling in some of these values and making this more robust when we're adding more and more data to it, so we can know and tell you all what's a kind of quote unquote a good or a poor or an outstanding soil health value based on your soil type. And then trying to tease apart a little bit more about, the management impacts. Okay. And that's again, a really important piece. All right. So I'm going to stop there, and we just have a couple more slides, folks, and we're going to turn to Q&A, but I'll just let Elizabeth take over here, and she can tell you where we're headed with some of this.
Thanks, Steve. So we're really looking to continue to integrate soil health measurements into more of our eFields trials. We're working on some funding to be able to incorporate yield and soybean cyst nematode measurements into this same project to be able to see how soil health affects other agronomic components in the field. We are looking again for some funding to continue to integrate the ability to incorporate some commercial labs into this testing to increase the speed at which we can return results, as well as make sure that our results that we're sharing with you are matching in parallel with what you would get from a commercial lab in Ohio. And again, like Steve has said, we've got a lot of data here to dig into, and we're really looking forward to better understanding soil health and how your management impacts the changes in those indicators and how that can help you guide decision making on your farm. So again, we're looking for funding, but if you're interested in getting involved, we're hoping to be able to continue this project this spring. The best ways to get in touch with us are, one, to contact your local extension educator in whatever county you reside in. If you would rather reach out to us directly, we can be found at the digitalag@osu.edu email address. Or if you're just interested in more general information about the program or to view the report, you can check that out on our website. And we're always trying to share what we're doing on social media. So in between webinars like this, you can check out what Steve is doing at the soilhealth.osu.edu website. All of the eFields as well as our other research is located at the digitalag.osu.edu website. And you can follow us on any of your social media platforms, YouTube, Twitter, Facebook, Instagram, to see what we're up to throughout the year. And so I think with that, we're ready to take any questions that you guys have.
Happy to take any questions. Yep.
Hi, this is Bridget, Moneymaker, another water quality associate. I have a few that I plucked from the chat that aren't in the Q&A box. One person was wondering, was the respiration lower for the manured fields? And was that expected?
Yeah, so let's take a look at that data slide real quick. So was respiration lower? I think, overall the distribution, again, because it's shifted right here, suggests that the respiration values were higher in manure fields. There's a taller peak here, which just means there's more observations right around this 45, 50 mark. That's where most of the non-manure observations fell out, where the ones, the fields that actually had received manure basically had a little bit wider range. And the distribution of those values is shifted right here. It's how we kind of read these graphs, that it's looking at kind of the overall distribution, not necessarily where that actual peak or point is. But yeah, I can see how that might be a little bit confusing because there is a taller peak here with the non-manure fields. But overall, what I take home from this graph and looking at this is that that when we have manure, a history of manure in those fields, it increases our respiration values, and that we know with our work that has implications in terms of more nutrient release with those fields relative to the ones that did not have manure. Yep.
Thanks. Another one that came through the chat, they were wondering about the influence of different types of fertilizer on that active carbon.
Yeah, it's a great question. We don't have really good information about that. There's been a lot of debate in the literature, in like science, over the last, say, 20 years, if fertilization, particularly nitrogen or phosphorus fertilization, either improve or increase total carbon or organic matter versus decrease, right? So we could go back to this slide here and thinking about this role of mineralization versus stabilization. So we know when we add nitrogen, it helps microbes speed up. It basically alleviates that nitrogen constraint in soil. And so microbes can chew up more carbon, more organic matter. But importantly, it also helps crops grow. That's why we fertilize, right? And so there was some debate about like if fertilizing, say with nitrogen or phosphorus, increased organic matter or decreased. And really, there's a lot of evidence to show that it actually improves. So fertilization, at least in my mind, fertilization means you have good crop growth. And that is really, really important because crop roots and residues are the foundation of carbon in the soil. It's the new source of carbon. That's what's driving all, we're talking about POX and respiration as carbon values. We want to see those increase. We need a lot of crop, healthy, strong crops for that to happen. And so my suspicion with judicious fertilization, we're going to help these numbers increase, although we really haven't looked at that directly. Yeah, hope that helps.
Another question for corn, what is an ideal carbon nitrogen ratio derived from healthy soil?
Yeah, soil total carbon and nitrogen, I guess we could take that question a few different ways, but total carbon and nitrogen in soil is actually fairly constrained. And so if we want to build organic matter, we need nitrogen to do it. Typically in soil, it's we can look across any soil in Ohio. Well, I shouldn't say any, but like if we exclude mucks, any mineral soil, nine carbons for one nitrogen. So it's a 9-to-1 to 12-to-1. That's really all we're going to find, say, in Ohio and across most of the mineral soils in the United States. And so, but just because, carbon nitrogen is very important as a concept, but it's very difficult to manage and try to, say, well, I'm shooting for a higher or lower. It's in my opinion too complicated to really kind of broad stroke it and say we really are shooting for this carbon nitrogen ratio for my soil type, right? So I don't really have a great answer aside from that. Yeah.
Thanks. So this one is a two-parter. First, do the soil testing practices use chemical extraction or water extraction? And then they're also wondering, these studies relate to organic practices. How might this relate to regenerative agriculture?
So I'll take the first question. I think that's probably talking about maybe some of these right here. And this is the phosphorus and potassium is a Mehlich-3 extraction. So it's not a water soluble or HA3 or whatever it is. It's not, that's just a kind of a classic Mehlich-3 extraction. Depending on I think that's what the question is about. And if I'm wrong, correct me. The second question was about organic versus regenerative ag. And, I don't have a good answer to that. Of course, organic, there's prescribed practices about, what you can and can't do. There's a list of OMRI approved products that you can use. Regenerative ag is more of recognize that there's a lot of overlap there, but it's a looser term in terms of what it means to different people. In general, regardless if it's certified organic, regardless if it's regen ag or not, the practices are the same. The concepts are the same. Keeping soil covered, roots pumping soil with carbon year round. That's really what we want, whether that be with perennials or cover crops, keeping that soil covered, maximizing diversity. These are the same kind of concepts regardless, right? And so that's the ultimate goal here, trying to fit that into a system and make money while we're doing it, right? Make that a sound economic piece. And how we get there, there's many, many paths to get there. And what we're showing here is just kind of being able to quantify that, taking those practices that are happening in the field, whatever they might be, cover crop, no-till. We all know, we're all aware of the suite that are out there. And being able to quantify this and give growers tools that they can track and see improvements and see how their management influences this. Okay, so anyways, that's a little bit of a long-winded answer, but I don't have a great answer to how organic versus regen ag would compare and contrast. I would think they would be mostly similar. Yeah.
Great. We have a few questions about manure, specifically what kind, liquid versus bedded pack, and the species. And then also someone's wondering if there's a difference between compost manure versus manure from storage.
Yeah, those are great questions. I don't think this analysis is really going to dig in that we just said, for example, was manure present or not. And so we know typically what type of manure they use, but we don't have great information in terms of rates and application. So we can ask for that information, but it requires a lot from growers to kind of document that. And we send these management surveys with farmers, and we try to keep it short and sweet. And even when we do that, it can be challenging. So anyways, and it's an unsatisfying answer, but we don't really have a lot of details to kind of tease apart all the different types and practices with manure. But we do have an unrelated manure project that we're working on, and we're hoping to provide a little bit more nuance and some understanding of that. Sorry, Bridget, I can't remember the second part of that question, actually.
It was wondering the difference between compost versus storage, so just more specifics.
Yeah. So more specifics, but of course we know that not all manure is the same, even though I'm treating it as such here. And, given that information, we could do a deeper dive and try to tease some of this apart. I don't think we really have enough data, enough numbers to really make, we start to split this data into, say, 4 or 5 different boxes, and then some of those boxes only have a few observations. It can be hard to really kind of tease that apart. Yep.
Great. So here's someone also asking about the soil test results, specifically at the 8-inch depth for P results. Were there any measurements of the corresponding soil pore water, so tile drainage, P concentration?
No, we didn't do any. I mean, I'll say this. We did, for this project, we didn't do any of that. It was just a simple one-time sampling. We are supporting, it's a, USDA ARS led effort for their edge of field studies when they're looking, those are paired comparisons across Ohio, mostly in the western part. That Kevin King at ARS and Will Osterholz are leading. We're providing soil health. We're still running those analysis, but in the early phases, that work is being done. It's really not being led by us, it's being led by USDA, but we're trying to link a little bit better soil health with some of the water quality piece. So we know that's an important story that needs to be told. It's very difficult really to quantify that and link those two things. Water quality on a field scale, soil health typically subfield scale, but trying to link those two things together is really an ambitious goal, but we're making a little bit of headway with that.
All right, and there are a few questions regarding cover crops and no-till, kind of a smattering of questions, but I think the overall theme is for soil health, specifically looking at tillage versus no tillage versus cover crops.
Mm-hmm.
Is anything specific coming out of that realm?
So that's a great question, right? So we look at this this thing and we say, okay, well, no-till, right? I'm not sure what that's happening. And then cover crops, we can see that but what if we looked at combine these two variables of no-till with cover crop and no-till without? A great question. Have not dove into that. Haven't looked at the data in that way, but it's something we really should do. The next step here is to really combine the 2 years of data and start to, because we have more observations and are able to do things like we're splitting, instead of splitting some of these practices up, we can start to do that. So that's something we'll definitely keep in mind and pursue. Thanks for that comment. Yep.
Great. I think we have time for one more quick question from the chat. It's a bit anecdotal. They're saying that they are seeing that adding nitrogen fertilizer increases bacterial respiration. So then you have to keep adding fertilizer to hold up mainly bacterial dominant microbial populations. Practices like cover crops promote more fungi populations, which demand less N and stabilize more N and C, and promote bacteria that can access more organic bound nutrients. Is this proving true in any of these studies or anything else that you've seen?
Yeah, I mean, great comments. And clearly, whoever wrote that has done their homework, and all, what they mentioned is true. Yeah, we, the tests that we've run here are really simple. Our respiration is just a 24-hour test. It's a kind of a simple one. So we're not able to look at things like bacterial respiration versus fungal respiration. So we don't really honestly have that much, we don't have a great comment about that. Like, we just didn't really look at it, don't have the ability to do that within the scale of this project. So, but, suffice it to say, we can look at the measurements that we did, right? And we can tease apart, and these are really measuring kind of what I would call a gross process. There's a whole bunch of microbes that are adding to, that are respiring, and whether those be bacteria or archaea or, multicellular organisms like nematodes and things like that. all adding to the pile of respiration. Okay. And so we're not going through and trying to tease out all the pieces here. That's really more into the research realm, right? Those are kind of more research-based projects. But if we take a step back, 1,000-foot view, at the end of the day, what we're trying to do is give growers tools that are quick, simple, but meaningful and reflect things. And so even though, and it's a good question, even though we're not teasing apart respiration here, we know that respiration in general, how much that soil respires, is very important. And we have to kind of pick our battles in terms of, do we want to do all this really detailed work that's expensive, or do we want kind of a simple, inexpensive indicator that can be ran and soils turned around? Because we have a soil health testing lab at Ohio State, but guess what, folks? We're slow. We don't have capacity. We're not a big dog like some of the soil commercial testing labs. And at the end of the day, the whole goal here, we do research. We don't, we're not a commercial lab. We want to push this out and get more commercial labs running this. And that's kind of the take home. So, that's our ultimate goal, is to get commercial labs up and running, and they can, turn numbers around quickly for y'all, because goodness knows it doesn't happen in our lab. We've got a lot of other things going on, research, et cetera. So, but anyways, I'll stop there, and much more than you needed in an answer, but that's what you got. Yeah.
Thank you, Steve, for that very detailed set of answers there. I'm sure that answered a lot of questions from our audience, and great presentations from both of you. Thank you so much, everyone, for attending today's webinar, and hopefully we will see you on March 3rd.